Hello my dear students, this is Dr. Pooja, your original guru of biochemistry. In this video of ours, we are going to discuss about lipoproteins. So let's start up with the discussion. Lipoproteins. Lipoproteins are a very important class of conjugate lipids. Now here if we say what are conjugate lipids, we know that lipid which is associated with a non-lipid part, which is known as the prosthetic group. And always remember, prosthetic group is different from the lipid component. So this prosthetic group here is a protein, right? And this protein which is associated to a lipid is known as a apoprotein. And this apoprotein, when it combines with a lipid, it is known as a lipoprotein. Right. So I can say that lipoproteins are formed when apoprotein combines with the lipid. Now the question is why? What is the rationale behind this? That why lipids always require a prosthetic group or a protein to be transported. So a very simple reason behind this is that lipids are non-polar in nature. We know that polar and non-polar, polar which are attracted towards water and which can be easily transported, non-polar which hate water basically. So lipids are non-polar, right? So they need a transporter to be carried in circulation. And if we compare carbohydrates and proteins with lipids, we know carbohydrates and proteins are strictly polar. So I can say that carbs and proteins being polar can be easily transported. So this is one reason why we require these lipoproteins easily transported. That is why lipids seek the help. It's like, just suppose if we have to travel from one distance to another distance, we cannot walk. So we are going to take another means of communication, may it a train, maybe it a bus or something like that. So lipids, whenever they move in circulation, they will always seek the help of these proteins. With the help of those proteins, they will be carried from one place to another, like from the intestines to adipose tissue, to the liver, to the brain, wherever they have to travel. The moment they enter in circulation, they require a protein. And just the protein component is known as a apoprotein. When this apoprotein combines with a lipid, it is known as a lipoprotein. Okay. And if we talk about the structure of a lipoprotein, obviously outer side will be of a protein and inner side will be of a lipid. So this is the protein component and this is the lipid component. This lipid can be, we can say, triglycerides or cholesterol esters. And suppose if we talk about phospholipids, because we have studied about phospholipids, so if I write a phospholipid here, you can say that phospholipid. So lipid is the non-polar part which will be inside and phospholipid will have a polar part will be outside. So I can say that polar part is always outside, right? And this non-polar part inside is inside. Similarly, if we talk about cholesterol, right? Cholesterol may be, I would say, the non-polar part will be inside and the polar part will be outside. Right, so this is how a lipoprotein is made and this is how these lipoproteins travel. Now the question is, what are the different type of lipoproteins? Right, and what is, because this charge and all matters a lot, because always keep one thing in mind, our cell membrane is selectively permeable. It will not allow any Tom, Dick and Harry to pass through it. It will be always selective in nature. Okay, so if we talk about the components, I can simply say that if we talk about triglycerides, always remember triglycerides are non-polar in nature. Very, very important. If we talk about phospholipids, phospholipids are amphipathic in nature. And if we talk about cholesterol, cholesterol is also amphipathic. Right? And if we talk about the proteins, protein here will be known as apoproteins. And this is very important. So the main thing is that triglycerides are always non-polar. Phospholipids and cholesterol are amphipathic. And if we talk about cholesterol esters, they will also depend. Why we have to make esters? There will be a change. So cholesterol is amphipathic, but ester is just to make it move. So it can be non-polar, it can be polar also. So depending upon again the composition. Okay. So depending upon this composition, there are basically 4 types of lipoproteins. Right. So if we talk about the type of lipoproteins, if we talk about the type, the first one is chylomicrons. Right. Second one is VLDL, which is also known as very low density lipoproteins, VLDL. Right. Third is low density lipoproteins. Fourth is HDL, heavy or high density lipoproteins. There is one more which we call as IDL. What is IDL? It is intermediary density lipoproteins. Okay, so this is known as VLDL, LDL, HDL and IDL. And chylomicrons are known as chylomicrons itself. So there are basically, you can say, 4 to 5 classes of types of lipoproteins. Now the question is, ek to why we have segregated them. Okay, and second important thing to remember here is, obviously there will be a difference in the composition of lipid or difference in the composition of the proteins. Okay, so what happens here is, always remember these chylomicrons are the biggest in size. It's like chylomicrons have this type of size, very bigger. Now, if chylomicrons have bigger size or the biggest, that means they are having more lipid content. So, they are having more lipid. Now, I will replace lipid by a clinical terminology, TGs. So, I can say more amount of TGs are there. And these TGs which we are getting from the diet. So, the TGs which we get from the diet, they are known as exogenous TGs or dietary TGs, whatever we can say. Dietary TGs or exogenous. Now, it is clear from the word itself if the size is more. Okay, the amount of protein required by that particular fat molecule would be less. So, it has least protein content. Protein content. So, if it has least protein content, that means its movement will be less. The more fat the person is, the lesser is the slowest is the movement. So, its movement will be slow. So movement is slow. Yes, slowest. And if we talk about the density, it will be least dense. Because density is basically due to the amount of protein present. Now, if we compare this with HDL, HDL is having this size, right? Now, HDL, this is having, if we talk about biggest size, this is having smallest size. Now, if it is having smallest size, first of all, we will talk about the movement. Movement will be more. Yes, यहाँ पे हमने क्या लिखा है? More TGs. It will have least TGs or in fact negligible TGs. It will have maximum protein content. So, if it has maximum protein content, least TGs. Right? And if we talk about the density, heavy density, that means maximum density. And it is also known as good cholesterol. HDL is actually good cholesterol. Why? Because it is responsible for reverse cholesterol transport, which we will be discussing in our video on the metabolism of HDL. So, here I am just writing this term, reverse cholesterol transport. So, I can simply conclude everything that density, it is inversely proportional to the amount of lipid content or fat content, or density is directly proportional to the protein content. This is very important. So, I will just highlight it also that density of a lipoprotein will be inversely proportional to the fat content and it will be directly proportional to the protein content. It means heavy density lipoprotein or HDL will have least fat content and it will have maximum protein content. On the other hand, chylomicrons will have maximum TGs, right? So, they will have least protein content. This is one very important aspect. Then second is, what are the methods by which we can separate these lipoproteins, because estimation of these lipoproteins is very, very important. Why? Because these chylomicrons, what happens is, if we talk about these chylomicrons, they have maximum exogenous TGs, Just keep this thing in mind. Exogenous TGs, what is the source of exogenous? It is the diet. On the other hand, VLDL. VLDL has maximum endogenous TGs. Now, if we talk about the source of endogenous TGs, it is due to the result of the metabolism which is occurring in our body. Maybe due to stress, maybe due to overexcretion of hormones, overproduction of hormones, it will lead to increased production of endogenous TGs. Exogenous TGs we are getting from the diet. Then if we talk about LDL, LDL is having maximum cholesterol. And that is why we always refer LDL as bad cholesterol. Then I mentioned about the fifth one, IDL. IDL is produced in between. When VLDL is being converted to LDL, IDL is produced. So basically IDL is nothing but it is a remnant VLDL, right? Or intermediary density lipoprotein. So we have to separate them. Now how we can separate them? There are two methods of separating these lipoproteins. Now what are the methods of separation of these lipoproteins? Methods of separation. There are two methods. One is electrophoretic separation or by electrophoresis, and second is by ultracentrifugation. So these are the two methods by which we can separate these lipoproteins. Now, if we talk about electrophoretic separation, we know what is electrophoresis? Electrophoresis. Phoresis is the movement. Electro means in the presence of electric current. So electrophoresis is the movement in the presence of electric current. Simple. And this is one of the very, very important methods of separation of proteins as per their size. Okay, here, because electrophoresis is of so many types, but here we are dealing only with the size. So if we talk about the size and if we talk about the movement, I can simply say that, say if this is the movement towards anode, the fraction which is going to move faster will be HDL. Then VLDL, then LDL, and then chylomicrons. And since HDL is moving at a faster rate, we will call HDL as alpha fraction. VLDL is behind that, so it is pre-beta fraction. LDL is more behind that, it is beta, and chylomicrons stay at the origin. So this is the movement, and that is why they are known as beta lipoproteins. And this is alpha. So alpha is the fastest moving fraction. So always remember, whenever we have to separate the lipoproteins by electrophoresis, HDL, which is actually a good cholesterol, so it will move faster. Why it will move faster? Because it has more protein content. And if we talk about the fat content, it is the least. And VLDL, I told you that HDL is responsible for reverse cholesterol transport. This is also very important to remember in this. Okay. So this is simple electrophoretic separation. Then second is ultracentrifugation. Now if we talk about ultracentrifugation, this is on the basis of the density. So ultracentrifugation में it is movement as per the density. Okay, so if we talk as per the density, so obviously HDL. So HDL is the one which will be like this, followed by low density lipoproteins. Here there will be a change, followed by VLDL, and then will come chylomicrons. Or we can say that chylomicrons will always stay at the origin, right? Like this. There is a difference because they are maximum dense and chylomicrons are least dense. Okay, so ultracentrifugation is a separation as per the density and electrophoresis is as per the protein content, or you can say as per the TGs, whatever we can say. Right. So these are the four main type of lipoproteins. No doubt IDL is also produced, but basically IDL is produced as an intermediary density lipoprotein, which we are going to cover in the metabolism also. So when VLDL is being converted to LDL, in between IDL will be produced. And it is also known as broad beta, also intermediary, or the other name of this is a remnant VLDL. And immediately it will be converted into LDL, then LDL has altogether a different metabolism to undergo. Okay. So these chylomicrons, or you can say all these lipoproteins, play a very, very important role. Use kya hai? Just to transport the lipids. Because when we are going to study the metabolism, we are going to study two types of metabolism. One is obviously the lipid metabolism, just like lipogenesis is there, lipolysis is there. But another segment which will play a very important role here, that will be the metabolism of lipoproteins. And that is why lipid metabolism is little bit vast also. Because here lipoprotein is an extra thing which is produced. And why it is produced? Again, to be transported, right? And clinically also evaluation of TGs is very important. Evaluation of HDL, evaluation of LDL, total cholesterol, these are the very, very important things which are clinically also very important. Okay, so you have to just understand this. So if I have to ask you the questions, if I say that maximum exogenous TGs are present in, always remember chylomicrons. Right, if I talk about maximum cholesterol, is in the LDL. In fact, LDL is the carrier of bad cholesterol. ये क्या करेगा? It will just take cholesterol and it will deposit it to the extrahepatic tissues, leading to a risk to the CVDs or CADs. Then, reverse cholesterol transport. HDL. Okay, and maximum endogenous TGs will be carried by VLDL, very low density lipoproteins. So these play a very, very important role when we are going to discuss about different type of hyperlipoproteinemias and hypolipoproteinemias. These are the ones which will be either increased or which will be either decreased. Right? So this is lipoprotein class, another very important class of conjugate lipids. So I hope this is clear to all of you, because we are going to study the metabolism of this in detail. So you should know what is an exogenous TGs, what is an endogenous TGs. It is clear from the word itself. Exo mane bahar se. So the TGs which are coming from outside. Outside means when we are taking a diet. So whenever if a person is showing maximum concentration of TGs, apart from medication, we are going to advise that person to have a diet which is low in the fats. And endogenous TGs are produced as a result of the metabolism in the body. And maximum endogenous concentration is there in very low density lipoproteins, and all will have a specific apoprotein. What is an apoprotein? Apoprotein is just the protein part. When this protein part mixes with a lipid, it becomes a lipoprotein. Right? So this is all about the video on these lipoproteins, which is a very, very important class of conjugate lipids. Thank you so much.